Thanks very much. I think, like all the other speakers, we're really honored to be here and sad all at the same time. I can say that David was just the most extraordinary colleague. Those of you who are in the field know that David really was a clinical immunologist. And in the United States, there aren't a large number of clinical immunologists. And so there was a natural bond, I think, that he and I formed, given that we were both looking at HIV, but really more through the prism of the immune system in many ways than the prism of virology or classic infectious diseases. Whoops. So what I'd like to do today is talk a little bit about the role of research in global health, and fully acknowledging that there is no chance that I would have been involved in this anywhere near to the way that I am hadn't been for my relationship with David and the ideas that he had, the ways that we might approach certain questions that we both felt needed to be addressed. And as you'll see, as I go through the talk, there's a certain evolution of that over time. We know about the disproportionate burden of poor health in less developed countries. But that's only a part of the picture, given that there are an enormous number of emerging and re-emerging infectious diseases occurring virtually on a daily basis. At the moment, for us, we're involved in looking at the more recent Ebola outbreak in the Democratic Republic of the Congo and the Nipah outbreak that appears, fortunately, to be subsided in southwestern India. But just looking over time, you have an appreciation for the large number of things that may occur, and just when you think you know what the next one will be, nature clearly is able to prove you wrong. There are a series of very important principles that I think guide studies in international clinical research. I think they were captured really quite well by Zeke Emmanuel, and I'm going to show a picture of Zeke and David a little bit later in the talk. Among those principles are that the research be done in a collaborative nature with collaborative partners, and those partners range from host country stakeholders, whether it's governments, academic institutions, affected communities, religious communities. The number of stakeholders and partners when one seeks to work outside of their own environment is very important to A, understand, and then B, fully engage. That the research have social value to the setting in which it's being conducted, that it be of high scientific quality, and that it be rigorous in its design and its execution, that just because there's an outbreak does not mean that the principles of science now can be discarded to try any investigational drug that someone has an idea might work. That the study population selected for study be done in a fair way that is consistent with the objectives of the trial, that there be a favorable risk-benefit ratio to the work, that it undergo independent review, that all participants provide informed consent, and that can be done in a number of ways. That's not just signing a form that outlines a whole series of risks that may or may not actually be applicable in a situation to theoretically cover the investigator. It's really making sure that the participant knows what the study is about and what their realistic chances are of benefiting or being harmed by the research, and that foremost there be respect for all participants in the research. I'm going to give a series of examples of global health research. For those of you who I know I've seen several of you already this morning who were involved back in some of the earliest work that we did with David, I'm going to bring back IL-2, which is something I know many of us haven't thought about probably in a long time. I'm trying to catch Sean's eye there, but I can't at the moment. And the two studies that were done there, ESPRIT and SILCAAT. Phidisa, which was a project we did in South Africa. The Insight Network, you heard a little bit of that from Sarah. I'll give you another perspective on that as well. And then finally, the last one is something that has not, at least to this point in time, involved the Kirby, but certainly has the opportunity to, which is some work that we've been doing in West Africa subsequent to the recent Ebola outbreak. So IL-2 actually is how I really came to know David. I had been at some advisory committee meetings, typically for drug companies when NIH employees were allowed to go there, which we still can go there, it's just on very different terms. And I would remember David getting there and sort of being in the room and really being so incredibly insightful and helping, which were predominantly a group of Americans around the table, look at things from a slightly different perspective. But I really didn't have the chance to work with him until this research opportunity came along. And we had been, this is work that goes to the mid-'80s, well, actually the early 80s. This was at a point in time, even some of this work before we even had the virus identified, looking at ways in which one might modulate the immune system to try to improve its function. And we had basically been trying anything and everything. If you had a cytokine, and we would try it, but they weren't really called cytokines then. They were just supernatants of stimulated lymphocytes. What we ended up finding, really quite fortuitously, was if you gave a patient 5 days of IL-2, waited 7 weeks, and gave another 5 days of IL-2, you could get these impressive increases in CD4 counts, as shown here on the top line of this graph. And that's in comparison, not a randomized comparison, to patients getting other treatments for HIV at the time. So you can date this slide by AZT-DDI, patients not on treatment, patients on single-agent AZT, or combination of DDI interferon, these being other studies that were actually ongoing at our clinic at the time. And to us, to look at this seemed really quite dramatic. And so the question was, okay, where would we go next to try to extend these findings? And as I mentioned earlier, it really seemed most logical to me to go to someone who had experience in clinical immunology, had that as their basis. The fact that it was, 7,000 miles away from Bethesda really didn't constrain me at all. So I said, really the next group we need to go to talk to about doing this would be David. And so here's a picture from the Daily Telegraph, June 6, 1994, talking about how we were going to develop and put together an international team of investigators to develop phase 3 trials of IL-2. I had no idea who to work with internationally. I had no idea where the different sites were that would be appropriate for something like this. But David had that just encyclopedic knowledge of places to work, individuals to work with, and really put together this list of 20 or so countries, 18 countries that we ended up working with in the phase 3 trials of IL-2. For those who weren't as intimately involved as I was or some of us in the room were, these really ended up being 2 different studies, one called ESPRIT, the other called SILCAAT. The ESPRIT study enrolled patients with CD4 counts above 300, SILCAAT counts of 50 to 299. In both studies, patients were randomized to either get combination antiretroviral therapy, and again, that evolved over time. So by the time of this study, we were talking typically about 3 drugs, 2 nukes, and a protease inhibitor or a non-nuke. Or just the combination antiretroviral therapy. Over 4,000 patients were enrolled in ESPRIT, about 1,900 were enrolled in SILCAAT. Again, the plan was to get 320 primary events. In ESPRIT, we ended up with 323. We didn't quite get the number we wanted in SILCAAT, ending up with 232, but between the 2 studies, a very large number of events. Even though the IL-2 treated groups had increases in their CD4 counts, shown here, 160 cell increase in ESPRIT, a 57 cell increase in SILCAAT, no impact on the viral load. Given this level of CD4 count increase, one expected about a 25 decrease in the incidence of serious AIDS-defining events or death. And what was observed was really a modest, non-statistically significant decrease. So in other words, even though these CD4 counts went up quite dramatically with IL-2, that did not provide any significant clinical benefit over what could be done at that time with antiretroviral agents alone. So interestingly, though, those sites that were identified to be part of ESPRIT, and I can even remember like it was yesterday, David and I sitting in a hotel lobby, actually in Johannesburg, talking about different countries, different investigators, people we then either singly, he or I, or the two of us together would visit, review the data, why we thought the study was something that should be done. Even though those studies didn't change the way patients with HIV infection were managed, they did actually create this network of people that really spanned, in essence, every continent. And it provided the stage and set the stage for what later became the Insight Network and the START and SMART studies. I'm going to come back to that in a minute, but just to try to keep things in the right chronologic order, I want to talk next about a project that we did that involved Sean and David in South Africa called Project Phidisa, Make Better, Prolong Lives. We've heard a lot about David as a leader, as a visionary, as someone who could get the job done. As I'll note in a moment, someone who actually knew how to have fun. I liked the picture Prapan from the HIVNAT meetings on a more casual moment, and it was great to get David on a casual moment. It really, I think, he helped all of us relax from time to time. In this setting, David really became a team player, and Sean in particular will know what I'm saying when I say, in this one, David took one for the team. So Phidisa was brought about by Thabo Mbeki's pronouncements that he wasn't really sure whether or not HIV caused AIDS, and maybe the drugs were making people sick, maybe poverty was making people sick, but he wasn't really sure that HIV caused AIDS. And many of you may remember just the amazing thing that was going on in South Africa at the time. Well, the interesting thing, if you think about a country, if there is a health policy that isn't quite aligned with reality and it affects your military, that can be a big problem. And that was what was happening in South Africa at the time. The general who was in charge of force preparedness, a guy named General Hadebe, had heard that HIV drugs actually helped, and that he wanted to get them for the soldiers because the sick bays were spending a lot of their time planning for funerals. So he went to the U.S. ambassador because he was told if you want to use these drugs, you have to do it in the context of research. He said, well, who do I go to for research? Someone said, naively, of course, the Americans. He went to the U.S. ambassador to South Africa and said, I'd like to do research. I would like to see if these drugs work. And the ambassador came to NIH. NIH ended up trickling down to me. I had been on the Mbeki panel debating whether or not HIV caused AIDS, indicated there was no way I was ever going back to South Africa to do anything, much less try to do research on treatment, until I went and visited with General Hadebe and his colleagues and realized how serious a problem this was. So I came back after a single visit and spoke with my leadership, my boss, Tony Fauci, Ed Tremont, who was heading the Office of AIDS Research at the time, Jack Whitescarver, who was the director of the Office of AIDS Research, and I said, no, this is a big problem. You know, we could do research there, but if you want to have an impact, it has to be big, it has to enroll a lot of patients, and this was sort of playing lessons from what we had done with ESPRIT, and I really need to put together a team of people who know exactly what they're doing, who can work well together. And among the first two people that came to me to put together as part of this team were David and Sean. Because of what we had done in ESPRIT, and also Jim Neaton and his colleagues at the University of Minnesota, because what they had done also with ESPRIT. So we put together this team. It involved the NIH, the DOD, University of New South Wales, and the South African National Defense Force. We designed a program to answer clinical research questions of relevance to both the US and South Africa. We had designed it to first look at HIV and then perhaps go to other areas. It really ended after HIV for a variety of reasons that were largely political. As I mentioned, it was started at the request of Ambassador Cameron Hume, and it did, I think, provide data that was important in seeing that health policy change in South Africa. One of the things we did in Phidisa that we've carried through in other projects of this type, particularly the ones that we've done internationally, is we parsed out, we had a governance so that eventually decisions would get made, and governance with the key stakeholders, but we parsed out all the different elements of clinical research, whether it was laboratory, pharmacy, protocol development, site management, ethics, training, communications. All of these different elements, we identified someone from the US or Australia to work with someone from South Africa. So that allowed us to, while we got the work done, also try to build the capacity. And Sean is over here helping with protocol development, and David is over here helping with training. Now, the reason David was helping with training was we had one person on the South African team who was convinced that you had to do a randomized placebo-controlled trial of ARVs to show that they worked. We took the position that we already knew they worked, and if that was what was going to be done, we would not be part of it. The person who was espousing that view was later assigned to the training group, and David was assigned to the training group. So David's role there was really using his great charm that we all know and his persuasive arguments to help bring that person in line with everything else that was going on. And I've always been very grateful to him for that work. That study set up 6 sites, 3 referral hospitals, 3 rural sick bays, just shown by the slides. It was a good example of the importance of community engagement. Here we have the South African Surgeon General at the time, von Rensburg, now the U.S. Ambassador to South Africa, and then 2 traditional healers during the launch of one of the 6 sites, this being the launch at Umtata. What the study showed was something that we all knew. We designed the study so that everyone got treatment. It was a 2x2 factorial, looking at different nucleoside regimens and then comparing an NRTI to a protease inhibitor. So those treatment regimens looked about the same at the end of the day. That was the primary endpoint. But among the data that were coming out were shown here. On the left, we're looking at the mortality rates for the patients we had enrolled in the study. We're following them, but hadn't had a chance yet to get them on therapy, compared to those patients who then got started on therapy. The lines are broken out by CD4 count. The red line, the top line, is CD4 counts less than 25. We were able to take these data to the Minister of Health in South Africa. We were able to take these data, coupled with the fact that the number of funerals at the sick bays were declining, to help provide some pretty clear evidence that the drugs indeed had an effect. And probably the only time any research I'm associated with would ever be in The Economist was this instance. So this is an article from The Economist talking about how in South Africa, in the public sector, it really is the military that was leading the way. And I do feel this project that David and Sean were key parts of had an input into the final decisions in South Africa to make ARVs available. Now, as I mentioned, David really helped the esprit de corps. I think only one of these wine glasses is his, though, Dory. I just want to point that out, that he always was a perfect gentleman and well-behaved. I can't say that for all of our colleagues who were in this project, but I can absolutely say that for David. And he's next to Zeke Emmanuel, who was the ethicist who had done those principles. Zeke also was the person who was one of the architects of what is affectionately known in the United States as Obamacare. So David would be the person, and you can see this look he's giving as the picture's being taken, who would always know the places to go and what to order from the menus. I have no idea how he did that. He knew better restaurants in Washington, D.C. than I did, and I've been in Washington, D.C. for 35 years. How he knew this, I don't know, but it was a gift. The next aspect of global research I want to mention is the Insight Network. You've heard a bit about that from Sarah, and this really, to me, exemplified David as a leader in global health. As I mentioned, some of the beginnings of Insight were from that ESPRIT study. And one of the components of ESPRIT was a group called the Community Programs for Clinical Research on AIDS, or the CPCRA. And you heard, just as a tragic coincidence, that Fred Gordon, who had been the head of that, actually passed away as well recently. In fact, Fred memorial service was Monday of this week, and at that time, many things were said about, Fred, his role with community engagement, and also his role with Insight. And it was interesting to me hearing the comments at Fred's memorial from the CPCRA people who are doing that. So for those of you who know some of the players, it was Wafa El Sadar, Donald Abrams, David Cohen, and they talked about how in the CPCRA, but it really applied to Insight, that it really was a team, and it was a team where the people cared about each other. So if someone had a problem, they weren't in competition with the other members of Insight. They really were there as partners, part of a family. Jim Neaton, who's the head of it, has a 3-word mantra: simple, flexible, fun, to try to determine, how we do. So Sarah, I would say that the study you talked about with 3 different randomizations, that would have been a hard Insight study to pull off. But other than that, the work in Insight was always geared towards trying to come up with better ways to treat patients, something that would have an impact on how patients were managed. And again, it really, combined that CPCRA with what had been ESPRIT. Now, CPCRA was doing a study at the time called SMART, and I'll just give a couple of slides on that in a moment, which was a strategy study, intermittent versus continuous antiretroviral therapy. But they were having trouble getting enrollment, and the study was a little bit at risk for that reason. So, with the help of David and others, and Jim, who had been involved in both, Jim Neaton been involved in both groups, the decision was made to merge what really was the CPCRA with the non-CPCRA elements of ESPRIT, and that really became Insight. Again, as Sarah mentioned, 2 of the prominent studies by that network were SMART and START. I'll talk about them both in just a moment. Dr. Fauci has often referred to those 2 studies as 2 of the 3 in the trifecta of HIV trials. One of the interesting things that was done with Insight, which had been done in ESPRIT, was really a distribution of authorities and management and responsibilities, such that there were 4 regional coordinating centers, Sydney being one, ICC's international coordinating centers. Minneapolis, Jim's University of Minnesota site, served as the coordinating center, and it really became a consortium that has been together ever since this time. The SMART study was a randomized controlled trial of patients with CD4 counts above 350, randomized to either a virologic suppression strategy, namely continuous antiretroviral therapy, versus a drug conservation strategy where patients were intermittently treated with antiretrovirals to maintain their CD4 count above a threshold of 350. The study was done at this time because there was concern that the drugs that were being used to treat HIV had significant toxicities that were showing up as cardiovascular disease, liver failure, renal insufficiency. The question was, what is the risk-benefit to these drugs? And if we could limit the exposure to the drugs yet maintain the immune system at a level of competency, could we have better clinical outcomes? Well, the study was stopped early because all the bad things were happening in the patients on the intermittent therapy. In other words, HIV infection was causing more than a state of immunocompromise. It was associated with a state of systemic inflammation, immune activation that had end organ damage consequences. This helped change much of our thinking about HIV as a disease. It has shaped some of the current research agenda today and opened up a whole area of basic science investigation as well, showing that in patients with HIV infection, certain inflammatory markers, in particular those reflective of a monocyte activation like IL-2 and soluble CD14, and those reflective of coagulation like D-dimer, have enormous odds ratios from the SMART study 6, 8, and 12, looking at the risk of death for those in the 4th quartile compared to those in the 1st quartile, or the highest levels versus the lowest levels. This was a very important finding to enhance our understanding of HIV. Second study by Insight, which really was the logical follow-on to SMART, was the START study, looking at when do you start antiretroviral therapy. Observational cohort studies had given some conflicting information. This randomized controlled trial, again, to either start therapy immediately in patients who are enrolled, the eligibility criteria was the CD4 count above 500, or initiate therapy once the CD4 count declines to 350 or lower. Again, this study was stopped early as there was a clear difference between the probability of a serious AIDS-related event, a serious non-AIDS-related event, or death, and those in whom therapy was deferred. This then provided really the final piece of definitive evidence from a randomized controlled trial that if you have HIV infection, you should be on therapy, and probably the earlier you're on therapy, the better. The last last project I just want to make mention of briefly is a project that's currently ongoing in Liberia called PREVAIL, which was part of the NIH response to the recent Ebola, or the 2014 to 2016 Ebola outbreak. I think you'll see as I tell you a little bit about this, that this project was actually modeled after the work of Insight, after the work of ESPRIT, after the work of Phidisa, after these projects that David had played such a central role in. This outbreak of Ebola from 2014 to 2016 in Guinea, Liberia, and Sierra Leone, by far the largest outbreak of Ebola virus that's been recorded. There were 28,603 cases, 11,301 deaths, 40% mortality. That 40% mortality is an interesting figure because if you read the textbooks at this time, that number would have been 90%. So the perception in the minds of the medical community was that this was a very, very serious disease, and not the 40% mortality is anything, you know, to ignore, but it's not 90%. And a lot of the decision-making about what research to be done was predicated on this notion of everyone's going to die, we need to try anything, as opposed to let's do rigorous randomized controlled trials to try to understand what's going on and make sure we make progress. And again, it's the rigor of the science that one does, I think, in any setting, whether it's an outbreak or in a chronic illness, that helps you build and go to better places to be able to provide better care. We were involved because of requests from the host countries, both Liberia and Sierra Leone, and then later Guinea, all sent requests to our Secretary of Health and Human Services at the time, Sylvia Burwell, asking if it would be possible to get some support to conduct research on promising therapeutics or vaccines for Ebola. We modeled this, as I mentioned before, after the way we had done Phidisa, with pairings of, in this case, U.S. subject matter experts with Liberian scientists to develop a clinical research program. The history of Liberia is a very sad one in many ways. A country that had been doing quite well and then was devastated by civil war, was just beginning to get back on its feet, and then devastated by Ebola. So in terms of the existing healthcare system, existing infrastructure for clinical research, it really was quite lacking. Again, having a clean governance going up to here to the Minister of Health and the U.S. Ambassador so we could get decisions made and not get bogged down in the politics. As I mentioned, we worked in the three hardest hit countries and Mali. Among the studies that were done initially were a randomized controlled trial of vaccines, a randomized controlled trial of treatment. That's been followed up now with a study of those who have survived Ebola, a genomic study, and most recently getting ready to initiate an HIV study. I just mentioned one of those studies, the PREVAIL 3, or the Survivor study. This is an observational cohort study that was started in June of 2015. It's goal to enroll about 1,500 survivors of Ebola virus disease and up to 6,000 of their close contacts. Again, the importance of controls in these types of settings, I just think cannot be overemphasized. The follow-up is planned for 5 years. We don't do a lot of laboratory testing. We do all the laboratory testing in Liberia, so we do serologies. We put in an eye clinic because of the risks of uveitis post-infection, detailed neurologic exams for the same reasons of concerns about long-term neurologic problems, and then serial testing of body fluids for the virus, or viral RNA. We had to go to some different ways of doing informed consent. You can see some picture boards up here. Here's the Liberian PI, Mosoka Fala, our lead for operations, Laura McNay, giving first a classroom session on what the study is about. That then is followed by one-on-one sessions. It's interesting that Dr. Fala had been involved in the outbreak response. He had developed the contact tracers, the people who were keeping in touch with those individuals who had been in contact with patients with Ebola. We pivoted those individuals to then help us with the study so that they help with the follow-up. And the follow-up on these studies is incredible. We have somewhere around 95 to 98% follow-up for each visit. So as an interim look, 1,000 survivors enrolled, 1,700 contacts. Of note, of the survivors, not all of them are antibody positive for Ebola, suggesting not some inability to make antibody, but a fairly significant number of misdiagnoses. And when you consider that when you have a diagnosis of Ebola, you go into an Ebola treatment unit, then you are potentially at risk for Ebola. This highlights one area needing improvement. Also of note, of the contacts who did not have Ebola, we found somewhere around 5 to 10% of them did have antibodies to Ebola. In other words, suggesting a spectrum of illness going from potentially asymptomatic all the way to life-threatening. Overall, the burden of illness was high in both survivors and close contacts. So there have been studies published on the consequences of Ebola virus infection looking at just the survivors and saying, well, there are all these medical problems. When you compare the survivors to the contacts who were antibody negative, most of those problems actually are seen in both populations and give you an idea of some of the healthcare needs within the country. We did see a higher prevalence of eye, musculoskeletal, and neurologic complications in survivors and controls. And interestingly, ourselves and others have found that a fraction of the men who are survivors still have elements of Ebola virus RNA in their semen. We're not sure what this means. Again, for Ebola virus, one of the real questions is where's the reservoir? What is the reservoir? This certainly suggests there could even be a human reservoir that may lead to new outbreaks. One of the reasons why we think it's important to follow this cohort for a period of time. So in summary, global health research provides an opportunity to accomplish something no single country can accomplish on their own and allows for direct investigation of many illnesses at the site of their greatest impact. To be successful, they must represent true partnerships among the participants, be driven by the public health needs of the populations they enroll, and have a clear governance structure. While local health policy and politics can greatly influence activities, the research must remain both ethically and scientifically sound. Successful programs have the ability to not only shape public health policy, but also to improve the capacity of local health systems and research infrastructures. And I just want to conclude by saying that David was a pioneer and leader in this area. His imprint on this area will indeed be one of his lasting legacies. Thank you very much.